This investigation is called looking into lactose, and it's mostly about enzymes. The reason we did this experiment was to identify the different kinds of milk and find the optimum temperatures and pH levels for lactase. Experiment 1 basically deals with identifying 3 milk samples. For this part, we inferred that if the 3 samples are tested, number 2 will be cow milk. We came to this hypothesis by using our senses such as sight and smell. The materials we used were 6 glucose test strips, a glucose concentration chart, 3 15 ml centrifuge tubes, a test tube rack, lactic solution, paper towels, lab aprons, goggles, bottles with the 3 milk types, 4 pipettes, and something to record the results in. And before we began, we made sure that safety came first. So, now for how to do the experiment. First, put 2 ml of milk from the bottle labeled number 1 into the first centrifuge tube. Then add some from bottle 2 to the next tube, and from number 3 to the third tube. Use separate pipettes for each one. After you're done with that, drip a glucose test strip into milk number 1 in the centrifuge tube, and lay it down on a paper towel with labeled sections. Now, do the same for the other 2 kinds of milk. Next, just wait 3 or so minutes, and then record the colors and concentrations using the given chart of the strips into the following table. Then gently mix the lactose solution and add 1 ml of it into each of the 3 tubes using a different pipette. Then cap the tubes and mix them gently. Replace the tubes into the rack, uncapping them. Dip another glucose strip into tube number 1 and lay it down on a labeled paper towel. Then do the same for the other 2 strips. Finally, just wait a few minutes and then record the colors and concentrations of the strips into this table. Before adding any lactase, none of the samples had any glucose. However, after adding it, sample number 2 contained a sufficient amount of glucose, while the other 2 samples didn't. This meaning that it was cow milk. Sample number 1 was rice, and sample number 2 ruled out to be soy milk. The color of the test strip before adding the lactase for each one was yellow, and the concentrations was 0. But after adding it, number 2 changed to dark green with a concentration of 1000. And here's a visual for all that data in a line graph. Sample 1 was rice, sample 2 was cow, and sample 3 was soy milk. In conclusion, our hypothesis was that if the 3 samples are tested, number 2 will be cow milk, and it was proven to be correct. This is because sample number 2 was the only one containing glucose, a sugar resulting from breaking down lactose. The experiment could be improved if more natural types of milk were tested, such as goat milk. This would have showed the experimenters that cow milk wasn't the only milk containing lactose. This experiment could be used, or is used, to educate people like us about the milk containing lactose, along with proving products such as lactics were successfully by scientists. So, on to experiment 2, determine the effect of pH level on lactase activity. Before we began, we inferred that if lactose is tested with different pH levels, then the optimum pH level will be 2 to 7. We arrived to this conclusion because as far as we knew, our bodies had multiple acids, but not many bases, thus meaning that most enzymes were made to work in these conditions. The things we used for this one were 6 glucose test strip, a glucose concentration chart, 6 15 ml centrifuge tube, a test tube rack, lactic solution, 12 ml cow milk, notebook paper, lab aprons, goggles, 6 different pH solutions, 2 test tubes, and something to record results in. First, add 1 ml of lactase into each of the 6 tubes. Then choose 6 different pH solutions, some acidic, some basic, and 1 neutral, and add 1 ml into different test tubes. Next, label a paper with your choices and then cap all the tubes and mix them gently. Then wait 5 minutes. Add 2 ml of cow milk to each test tube, then cap and mix again. Dip separate glucose strips into each solution and lay them down on the labeled notebook paper. Then wait 60 seconds and record the colors of the strips in a table. Finally, just record the glucose concentrations using the chart. So here's our findings. For pH 7, the color was green, pH 2 dark green, pH 12 yellow, pH 8 green, pH 6 dark green, pH 3 dark green. And for the concentrations, we got pH 7 1000, pH 2 3000, pH 12 0, pH 8 1000, pH 6 3000, and pH 3 3000. And here's a bar graph. To basically summarize our results, high glucose concentrations were found in pH 2 to 8. This means that from level 2 to 8 is when the enzymes function successfully because the job of it is to convert lactose to glucose. The more basic the environment is, the more limited the enzyme's function is. This is because high pH levels affect the enzyme's shape and properties. So we concluded that our hypothesis was, if lactose is tested with different pH levels, then the optimum pH level will be 2 to 7. And was partly correct, because our results said that the success rate was from 2 to 8, meaning the more acidic the environment, the better the enzyme's ability to function. This experiment would have been much more accurate if all of the pH levels were tested. It would give us a whole picture of things. This experiment could later be used or is already being used to prove the quality of the product called lactates. In experiment 3, we found out what temperature worked best for lactates. The materials we used were 6 centrifuge tubes, a test tube rack, something to pick up the tubes, lactate solution, milk, a thermometer, 3 glucose test strips, something to record results in, 2 pipettes, beakers with water, hot plate, and a beaker with some ice. We predicted that if the lactose is tested in 3 temperatures, it will work best at room temperature. We arrived to this because the enzyme was probably created at room temperature, meaning that that was the temperature it was made to work best in. Also, in previous experiments, we used milk at room temperature, and the enzyme worked fine then. Now for the procedure. First, mix the lactate solution and then add 1 ml of it to the tubes ending in L using a pipette. Next, add 2 ml of milk to the test tubes ending in M. After that, cap all the test tubes except the one starting with H. Now, put the tubes in their matching temperatures. For example, hot and water bath. Then use a thermometer to record the temperatures in the following table. Pour the contents of the milk tube into the corresponding lactate tubes. Example, RM to RL. 7. Cap all the tubes and mix them gently. Then put the tubes back into their respective temperatures for 5 minutes, leaving the hot ones open. Finally, test the solutions using a separate glucose strips, and then record the colors and concentrations in the table. At 20 degrees Celsius, the color of the strip turned green with a concentration of 1000. At room temperature, meaning 74.4 degrees Celsius, the strip turned dark green with a concentration of 30,000. While in hot water, meaning 101 degrees Celsius, it turned light green with a concentration of 100. And here's a bar graph showing a visual of all that information. The enzyme worked best at room temperature with a concentration of 3000, and the second best in cold with a concentration of 1000. It didn't work so well in hot temperature though, with a concentration of 300. The room temperature was the best place for the enzyme, as its property and such weren't affected by the temperatures. Our hypothesis was that if the lactose is tested in the 3 temperatures, it will work best at room temperature, and it was proven to be correct. This is because the color of this glucose strip that tested room temperature held a concentration of 3000, far more than any of the others. This experiment could be improved upon if a wider variety of temperatures were tested. For example, almost frozen cold and super hot. This experiment could also be used by scientists or everyday people just to find out the optimum temperature ranges for other artificial enzymes. So that was it. Wait, there's some credits. We hope you enjoyed our presentation. Thank you.